So I'm excited to tell you today about some of our recent work on plant microbiome interactions, and hopefully with this longer format, give you a little extra background about the field. Feel free to interrupt with questions in the chat or raising your hand, and I can stick around at the end for a while to answer questions as well. So when I think about life and agriculture on this planet, I think there's two chemical reactions that are critical for life as we know it, really. So the first is nitrogen fixation, where microbes can take nitrogen gas from the atmosphere and pull apart that really strong triple bond and convert it to ammonia. So this is a biologically available form that's critical for life. The second is carbon fixation from plants and microbes that can take carbon dioxide from the air and convert it to sugars. So both of these are central for life, and without these processes, we wouldn't have biologically available nitrogen and sugars. So the most critical evolutionary innovation, I think, for agriculture and for feeding people and other mammals was when plants and microbes joined forces for symbiotic nitrogen fixation. And so this is when plants, which photosynthesize, form a mutualistic symbiotic association with rhizobia bacteria. Plants make these specialized structures called nodules on the roots, and within these nodules, plants and bacteria exchange carbon and nitrogen. And so, as I'm sure you all know, this has really been critical for fueling agriculture. And it's been critical for as long as agriculture and humanity have been around. So as early as the 5th century BC, Romans realized that alternating beans and peas with other crops could enhance productivity. And so this has been used for centuries to fuel agricultural productivity. However, symbiotic nitrogen fixation, as you all sure know, is limited to legumes. And so a question that's been central to plant microbe interaction research is how can we harness this chemistry for other plants? So the first Green Revolution was focused on answering this question. In the early 1900s, the German government saw that there was a potential population and food crisis, and so they invested in agriculture. And two chemists, Fritz Haber and Karl Bosch, discovered an artificial nitrogen fixation method to actually generate ammonium from atmospheric nitrogen. And so this is what we call the first Green Revolution. And they discovered that by taking nitrogen gas and mixing it with hydrogen in a compressor under tremendous heat and pressure, they could pull apart that triple bond in dinitrogen gas and make ammonia. And so this has been probably arguably the most important innovation in agricultural history. This is a plot of corn yields over time in the United States. And you can see with the application of nitrogen fertilizer, which occurred about here where the arrow is, corn yields skyrocketed. And so estimates are that between 30 to 80% of the nitrogen atoms in your body originated from chemical nitrogen fixation. And estimates are that we could only feed about 4 billion people on the planet without it. And so about half of the people on the planet are only here. I just got muted. About half of the people on the planet are only here because of chemical nitrogen fixation. And a little side note, Fritz Haber is a fascinating character. And so if you're interested in this, I would definitely recommend Googling him and reading more about the history. He's one of, I think, science's greatest heroes and also greatest villains. So there's some really interesting stories there that I won't get into now. So despite the importance of chemical nitrogen fixation and fertilizer, as you all know, this is economically and ecologically extremely expensive. So in Canada, which is a relatively small agricultural country where I live, we put about 5 million tons of nitrogen fertilizer onto our crops each year, and this costs us about $4 billion. And so scaled globally, you can only imagine how expensive this is. And less than half of this fertilizer is actually incorporated into plant tissue. The rest remains in the environment, so it can run off into lakes and streams, and it can actually damage ecosystems. So this is quite both economically expensive, are currently failing to meet global food needs. So it's estimated that in the next 30 years, we're going to reach 9 billion people on the planet. And at the same time, about 1 in 8 people are currently undernourished. This problem is only going to get worse. So these are projected yield changes in the next 30 years, and particularly the Southern Hemisphere. We're projected to have potential decreases up to 50% due to decreased rainfall, increased temperature, and increased pathogen ranges. This is less of an issue for us here in Canada and you in Northern Europe, where warming climates might actually help agricultural productivity. But currently, the people on the planet that are already limited by calories, nutrition, and food security are going to be further impacted. And so not only that, our crops need more than nitrogen. So in the face of global warming, we have increased pest ranges. So insects are spreading farther and into new environments. Our plants are having decreased nutrition due to elevated CO2. So more fixed carbon means that proportionately we have less of other nutrients in our plants. We're having increased drought due to decreased rainfall and increased temperature. And milder winters are not suppressing pathogens. So not only are they spreading into new ranges, but they're actually becoming worse in their local environments. And so these are challenges that we need to address to ensure food security going forward. So we now, I would argue, need a second green revolution that meets global food demands, that addresses climate change, that does this sustainably and affordably. Because we really need to get this technology to the people that need it most. So I think part of the solution can lie on our crops as they already are, and these are the microbes. So microbes coat every surface. Not only are they in nitrogen-fixing nodules and legumes, they're on every root surface, every leaf surface, fruit, and flowers. And these microbes provide a lot of benefits to plants. So they can help with agronomically important traits like flowering time and yield. They can help with nutrient availability, such as nitrogen fixation, but also other micronutrients in the soil. They can help with adaptation to climate change, and they can promote disease and pest resistance, both by antagonizing pathogens and by modulating the plant immune system. And so many of these challenges from global warming can potentially be addressed by microbiome engineering. So microbes can increase resistance to insects by actually making insecticidal compounds or modulating the plant immune system. They can help with increase of micronutrients, which can help with decreased nutrition. They can improve plant drought tolerance and salinity tolerance. And they have antimicrobial activity that can kill fungal and bacterial pathogens. So today I'm going to tell you a little bit about the current use of microbes and microbial traits in agriculture. And then what we know and what we're good at in terms of using microbes for agriculture, and then where we still need to learn more. And then I'm going to tell you a little bit about the reductionist approach that my lab and many other microbiome labs use and sort of the rationale for that. And then I'm going to tell you 2 stories from our lab, how microbes can provide the benefits that they do to their plant hosts, and then how the host itself can shape this microbiome community into one that's beneficial. So there's the idea for microbiome engineering is that we can, in a rational way, take beneficial microbes from roots or other plant surfaces and then add them to plants that may be lacking these. And plants may be lacking beneficial microbes because they live in disturbed soils from agriculture. It's possible that the microbes just weren't there in the first place, or plants just need an extra boost that they wouldn't have gotten sort of evolutionarily. And so we need to actually engineer the communities into one that's more beneficial. And so this can come in the form as single PGPR, as plant growth promoting rhizobacteria, or other. This can include other microbes with beneficial functions. So this can include single inoculum. It could potentially include a synthetic community where you put several microbes that could work synergistically to benefit plants, or potentially soil transplants. So you could take soil and take a microbial extract from it and transplant it to plants. So theoretically, these are the approaches and strategies we're thinking about for engineering the plant microbiome. So in reality, there's actually only a few limited uses, and they tend to be in this single microbe inoculum. And so a classic one that I'm sure many of you know is Bacillus thuringiensis, or Bt toxin, which is an insecticidal bacterium. So Bacillus makes this Bt toxin that kills insects, and it works through adding spores to soil or plant leaves. And it can confer resistance to a variety of insect pests, shouldn't be pathogens. Plant varieties can also be engineered to produce Bt toxin, and so this is widely used in North America as a way to protect against insect pests. And Bt is arguably, in terms of insecticidal microbes, it's arguably the most effective. It really works, and it likely works and is so versatile because we know the genes that make Bt toxin. And so we can either use spores and spray them on plants for organic agriculture, or you can actually express heterologously Bt toxin in yeast or other vectors to purify and then spray on plants, or we can engineer plants with those genes. And so not only is this effective, it's quite versatile and can be used for a really diverse array of agricultural applications. And so this is really, when we're thinking about single microbial inoculants, this is really kind of the gold standard because we know the mode of action and we can use it in all of these different versatile ways. The other common use of microbes is sort of similar in that they're used as post-harvest fungicides. And so for this, bacteria can coat the surface of fruit or leaves and kill fungal spores as they potentially enter wounds or other vulnerable locations on our fruit. And there's many bacteria that have known antifungal traits. And in some cases, again, the mode of action are known. And again, these are quite effective to be used as post-harvest fungicides. And there's also several that are approved for foliar application in the field. And so this is, in these cases, bacteria are like Bt, being used in a similar manner as fungicides, and they're coating leaf surfaces or fruit surfaces. And so here we're basically using microbes as a delivery vehicle for fungicides or pesticides. And so the cases where microbes work tend to be biocontrol of pests and pathogens. So when the mode of action is a single genetic trait, the molecule can be isolated, or when we apply them as foliar applicants or fruit applicants. So there's reduced competition, and I'll come to that with the soil, when you're applying these things as fruit or foliar applicants. Where we really don't see microbes being used is a lot of the area that I described as potentially combating challenges for climate change. So other than rhizobia, no bacterial soil treatments are really used on a large scale. And microbes that show success in the greenhouse often fail in the field. And so we'll find a really promising microbe in the lab, we'll test it in the greenhouse or maybe even a local field. But then when this is expanded to more fields, we find that they don't work. And so why are they failing in the field? We think that it's likely because it's linked with plant colonization. The traits may be polygenic. So when you're thinking about something like growth promotion or nutrient uptake, it could be that there's more than one gene that underlies the process. And it may be that it really needs to be adapted to the local soil and climate. And I think this has really been underappreciated for microbes, right? We think about adaptation of cultivars of crops to a local climate, but people often will take a microbe that's been developed, in a very hot tropical climate and then try it in Vancouver where it's cool and wet and it doesn't work. And I think that's like trying to transplant a cactus into Vancouver. It's not necessarily adapted to our local climate. And so I think all of these things, when we want to actually engineer soil microbiomes, it's going to be critical to understand how they colonize plants, how the plant genotype shapes the community, the genetic basis of these traits, and then how the local environment actually is important for microbial adaptation and how microbes can survive different soil conditions and different environments. And so one of the major goals of my lab is to understand first what makes individual bacterial strains or a community beneficial, so finding the genetic basis of these processes, and then how the plant shapes the community. So if we put a microbe on a plant, will it be able to survive or not, and what determines that? So To achieve this, I think we really need reductionist models. And so I'm going to tell you a little bit more about the rationale for that and the approach that my lab uses. So first, I'm going to tell you a little bit more about the rhizosphere microbiome. So my lab works on the communities of root-associated microbes, and these are incredibly complicated. So if we look at a gram of soil, there's about 10 to the 6th bacteria per gram, so 1 million bacteria per gram. And within that gram of soil, we can have up to 1 million different species of bacteria. So OTUs are operational taxonomic units, and this is our metric of bacterial species identity. And so there can be up to 1 million unique bacteria in a gram of soil. If we look at a root, there's actually more bacteria, so usually about 100 times more bacteria per gram of root, and that's because plants take photosynthate and they secrete it into the soil. Estimates are that 10 to 30% of fixed carbon from plants actually goes into the rhizosphere, and so you can almost think of this like a nectary for bacteria. So plants are feeding the bacteria through this fixed carbon. However, the diversity of microbes is quite reduced compared to soil. So we have more like on the order of 1,000 or maybe 10,000 bacteria, depending on if you're on the root surface or inside the root. And this is because the plant immune system plays a gatekeeping role to restrict microbial access. So to compete along the root, bacteria need to be able to use plant-derived sugars, and they need to be able to compete with this community of microbes. And they also need to survive the plant immune system. And so there's a lot of metabolites plants make to restrict access for a lot of these microbes. All this being said, this process is genetically controlled by plants. So if you take the same plant and you put it in the same soil, you largely get the same community out. So the soil is the largest determinant. The million bacteria that are there to start with are going to determine that final community. But there is a plant-driven program that results in a consistent community forming around the plant roots. So a great thing for research is that despite the complexity of these communities, if we take a germ-free plant and we put a single bacterial strain, it's often sufficient to confer benefits. It's actually remarkable how many bacterial strains we can isolate from a plant root and then add back to germ-free plants and then see some effect, whether it's nutrient uptake or growth promotion. And fortunately, many of these are model bacteria. So things like Pseudomonas and Bacillus and Streptomyces that we've studied in the lab for their ability to form spores or make antibiotics or form biofilm turned out to be naturally plant associated. And we have genetic tools for these microbes. So the microbial community is incredibly complicated. And when I try to think about what each individual is doing, it's like trying for me to tease apart the individual contributions of members of a symphony. It's incredibly hard to isolate what each member is doing. And so, yeah, I guess I just get overwhelmed. And so when I think about how to approach this we've taken a reductionist approach, which is where you try to focus in on what a single member is actually doing and take that individual in isolation, or even the microbes on the back of a hand of a member of the symphony to try to understand the conversation between two partners. And so the goal is to try to actually isolate what each individual is contributing to this conversation, and then maybe piece the whole thing back together. And so to create the symphony, we need to understand, or each player needs to know what they're contributing. And so that's sort of the rationale for a reductionist model, where we take a tractable plant-microbe interaction, and robust phenotypes are critical for this, and I'll come back to that, to try to uncover mechanisms. And so this is something that we kind of intuitively know from the plant-microbe interaction field. And so finding mechanisms in our field has been driven by using model reductionist systems. And so this is true for pathogenesis, where we use a single plant interaction with a pathogenic microbe, and we can get things like hypersensitive response, and then we can screen for a mutant that loses hypersensitive response. And that's been the basis of finding things like plant disease resistance genes. In nitrogen-fixing rhizobia legumes, one of the powers of that system and why it's been a model for symbiosis is that, again, we can have this robust phenotype of inability to fix nitrogen in nutrient-poor, nitrogen-poor soil that's allowed us to screen for plant and microbial mutants and then study them to find things like nod factor and the plant receptors for nod factor. However, in the microbiome field, this has been a lot more difficult because we kind of lack the robust phenotypes, or intuitively lack the robust phenotypes of pathogenesis and mutualism. And so things like plant growth promotion result in maybe a 10% decrease in growth of plants. So they're relatively subtle compared to something like nitrogen fixation, where it's this really stunted, sad plant in nitrogen-poor soil. And so these become very labor intensive to screen for. And I think that's really slowed down progress in our field, just a lack of robust phenotypes to allow for forward genetic screening. So the past couple of years have seen what I would call closer to more forward genetic screens, and people have really started looking for more robust phenotypes to screen for. But I think, in general, we're taking a long time to catch up to immunity and symbiosis fields because of the difficulty of genetic screening, but I really think that's critical. And so to develop a system where we could actually start doing forward genetic screening, my lab and most or many other microbiome labs that work on mechanistic questions use the model plant Arabidopsis for all of the reasons that it's used for pathogenesis research. It's an excellent system for forward and reverse genetics. It's our validated reference plant. So what we've learned about immunity in Arabidopsis and flowering time has been largely translatable to ecologically and agronomically important plants. Importantly, the Arabidopsis microbiome community, at least at a coarse phylum level structure, resembles that of crops. And so this is a plot showing phylum level diversity of Arabidopsis, corn, and rice. And so you can see that the colors look sort of similar. And so the community structure of Arabidopsis is, at least at this taxonomic level, similar to some of our important crop plants. And then really importantly, Arabidopsis is a wild plant. It's not domesticated. And so for many of our crops, we have potentially lost things like disease resistance genes, right? Some of the first R genes in tomato were just natural or loss of function through domestication. Whereas Arabidopsis is a wild plant that's been allowed to co-evolve with its microbes and its pathogens. And so while we've potentially lost some of genetic diversity for disease resistance and potentially microbiome associations in our crops, if they haven't been selected for, Arabidopsis retains those associations because it's a wild plant. My lab studies the interactions of Arabidopsis with diverse Pseudomonas species as a model to understand plant microbiome interactions. So we primarily focus on Pseudomonas fluorescens and related species because they're enriched in the plant rhizosphere microbiome, usually 10 to 1,000-fold relative to bulk soil. And they provide a lot of diverse benefits to plants, so they can induce systemic resistance or antagonize microbes. They can also promote growth. They're really interesting to study from a microbiome perspective because their function can't be inferred from taxonomy. So that 16S ribosomal RNA metric that we use for bacterial diversity, so the sequence there tells us bacterial genus level identity. You can't infer function of Pseudomonas from 16S. And then Pseudomonas is tractable with thousands of sequenced genomes. And so we have a number of tools to actually approach genetics of Pseudomonas plant interactions. So the genus Pseudomonas, I think, is fascinating. It has a huge amount of functional diversity. We have Pseudomonas aeruginosa, which are opportunistic human pathogens and can even be pathogens of plants. We have Pseudomonas syringae, which is probably the most well-known Pseudomonas to this group. So these are plant pathogens, and they're also model plant pathogens. And then there's Pseudomonas putida and Pseudomonas fluorescens, which are commensals of plants and bacteria, plants and animals. And my group primarily focuses on Pseudomonas fluorescens. However, I think this genus is really fascinating because we have so many different host-associated strains. So they seem really good at just being host-associated. And then there's a really huge diversity of lifestyles of Pseudomonas. So we have mutualists, we have pathogens, and we have commensals that neither harm nor benefit their host. And so I think Pseudomonas is also really interesting for just studying the origin of host association. So within Pseudomonas fluorescens, so within what we would call a single species, or 97% identical by 16S ribosomal RNA, first we have thousands of genomes, and so this makes Pseudomonas a really excellent tool. And we also have a huge variety of functional consequences for plants. And so not all strains do all of these things, but individual strains can do things like protect plants from herbivores. They can help with plant nutrition, so they can help with nutrient uptake by solubilizing things like zinc and iron from the soil. They can induce systemic resistance, where they make plants more resistant or susceptible to pathogens. They make antifungal and antibacterial compounds that can, this is nutrient solubilization. They can make antifungal compounds that kill fungi directly. And as I mentioned, they can solubilize, this is zinc. And they can promote plant growth or change plant root architecture. And there's even evidence of opportunistic pathogens in this group, which I'll tell you about in just a minute. And so I hope I've sort of set up our use of reductionist plant microbiome models and the need for using them to tease apart the complexity of these communities. So now I'm going to talk about two projects in our lab that are fairly recent. First, how microbes provide benefits to their plant hosts, and then how the plant actually shapes microbial communities. So we're really interested in understanding how Pseudomonas and other bacteria provide all of these benefits to their plant hosts. So to do that with a photogenetic approach, like you would approach a question like how does a plant perceive an effector, you would make bacterial mutants or plant mutants, and then you would screen for impaired interactions. And so if we want to ask a simple question in the plant microbiome field, what's the genetic basis of growth promotion, for instance, you would take your bacterial mutants, add it to a plant, and then ask if the plant is bigger or not. However, these are relatively difficult phenotypes to screen for. You can't, like hypersensitive response, screen in a single individual and get a good readout of whether it's happening or not. Here you have to weigh many plants because it's, as I mentioned, on average, you just get a slight boost in plant size. Or if you're screening for induced systemic resistance, again, you have to measure many individuals because the phenotypes can be quite subtle. And so these are very labor intensive to the point that I and others have failed to convince graduate students to take these projects on. So they're hard to screen for to the point that this approach has really been limiting in our field. So instead, an approach that our lab and other labs have used is to try to make use of the natural variation in bacteria. So two strains of Pseudomonas fluorescens, so the same species, may only share 50 to 90% of their genetic content. And this is really different from eukaryotes. So if you're used to thinking about diversity in Arabidopsis or even in primates, right, we share 99% of our protein encoding gene content with our nearest primate relatives. And so if you compare that to two Pseudomonas strains that may even be the same species, they may only share 50% of their gene content. And so we can take advantage of this because if we now just compare two strains and we see that strain A promotes growth and strain B does not, we can look for genes that are unique in strain A that might actually contribute to that process. And so Pseudomonas has about 6,000 genes, only 3,000 are components of the core genome. And so as we add more strains to this, we can start to narrow down these unique strains to unique genes to the growth promoting strains. And so if you do this comparison of two, you might find 1,500 genes that are unique to the growth promoting strains. But if you add another strain that's diverse, you might narrow this down to 750 and so on. So with about a dozen strains, we can narrow this down to a reasonable number of genes that gives us hypotheses to test. And so I'm going to talk about the work of a former postdoc in the lab, Ryan Melnick, who tried to use this approach to answer a really basic question in our field: what's the genetic basis of growth promotion? And so he wanted to use one of our lab's favorite strains, which is the Pseudomonas WCS365. And it's a really robust growth promoting strain. And in this case, we don't know the basis of growth promotion by this strain. And just to orient you, the Pseudomonas fluorescens species is 97% identical by 16S ribosomal RNA. And Ryan started within the Pseudomonas brassicas subclade, which has 99% identical by 16S ribosomal RNA. So these are very closely related bacteria. And so what Ryan did was just ask, how far do you have to get away from the strain to lose growth promoting abilities? And then can we use comparative genomics, compare the growth promoting strain to the non-growth promoting strains to find the genetic basis of this process? And to our surprise, we didn't actually have to get very far. This N2C3 strain here, which is 99% identical to our growth promoting strain WCS365 and N2E2 shown here, it's actually a pathogen. So here's in germ-free conditions, control buffer-treated plants. If you add with N2E2, you actually don't get huge amount of growth promotion because this is on nutrient-rich media. And so again, the problem with growth promotion, you can see these changes in lateral root architecture, and then under nutrient-poor conditions, this promotes growth more robustly. But under these conditions, N2C3, this strain here, stunts plants. And so this is just death quantified. You can see that this strain severely stunts plant growth. It turns out it's not unique to Arabidopsis. So this is N2C3 on rice, and you can see it prevents seed germination and kills rice as well. And it turns out that it kills almost every plant that we've tested. So we just sort of screen through all of the seeds people had to plant in their garden in the lab, and it kills poppies and tomatoes and rice as well. And so this seems to be a broad host range pathogen. And so we used a comparative genomics tool that was built by Ryan, and this is available on our lab's GitHub. This has been really useful for our lab because it turns what is potentially a computationally exponentially difficult problem into a linear one. And the way this works is normally if you would want to compare all the genomes of different strains, you would do all by all comparisons, which becomes computationally exponentially difficult. Every time you add a new strain, you have to compare it to everything else. And so this takes a lot of processor time. So what Ryan did instead was took bacterial genomes that were high quality, so finished bacterial genomes, and he used those to generate orthology groups. And then he generated a single giant pangenome for Pseudomonas. So this has about 20,000 orthology groups that represent all of the genes available in Pseudomonas. And now we can compare each individual strain to that single pangenome. And so it turns this into a computationally linear problem. And so now this is something that you can just run on your computer, on your local laptop computer. And we've expanded and worked with a number of groups to implement this for other well-sampled bacteria like Vibrio or Bacillus. And so this is available if you're interested in applying this to your own bacterial group. So Ryan used this as a test case to try to find the genetic basis of this pathogenesis phenotype. And so he looked for genes that were unique to the pathogen and absent in the commensal. And he also potentially looked for genes that were unique to the commensals and absent in the pathogens. And I'll come back to that later. So in this particular case, it turned out to be something quite obvious that maybe we didn't need such a sophisticated tool for, but this tool has been useful for many other applications. It turns out that N2C3 has an island encoding lipopeptides, syringomycin and syringopeptin, inserted in its genome in a location that's otherwise perfectly conserved with N2E2 and all the other commensals. So this is located here. It's a very large island, 143 kb, and it has a quorum sensing regulator driving syringomycin and syringopeptin, which get their names because they were first characterized in Pseudomonas syringae. And so these encode toxins that are well known to be responsible for virulence. And so this is Pseudomonas syringae on bean, and this is one on cherry. And then here's mutants, where people have deleted the syringomycin and syringopeptin production genes, and you lose virulence of these pathogens. And so they're well studied, reasonably well studied in Pseudomonas syringae. So this is a type 3 independent virulence mechanism. And so Ryan deleted syringomycin and syringopeptin in N2C3 and found that they actually work synergistically to kill plants. So if you lose either of them, you actually restore plant growth. And then if you lose both, you actually restore plant growth even more and to similar levels as the non-pathogenic strains. It turns out that this island is found broadly within the genus Pseudomonas and other generalist plant pathogens where the virulence mechanism hadn't been previously described. And so this is Pseudomonas fuscovaginae, which if you look up here, Pseudomonas syringae, there's some strains. So DC3000 doesn't have syringomycin, but there's other Pseudomonas syringae strains that have it. And then these fuscovaginae strains are relatively closely related to syringae and are generalist pathogens. And so these cause rice sheath rot. And then this is Pseudomonas fluorescens. And then this is our Brassica sciarum clade. And within this group, there's actually known generalist pathogens like Pseudomonas corrugata that causes tomato pith necrosis. And this also has syringomycin. And so it looks like this may be horizontally transferred from pathogens into these commensals and is the basis of many broad host range pathogens. So zooming in on just this Brassica sciarum clade, so just this group here that's 99% identical by 16S, it turns out that this islands, including syringomycin, syringopeptin, has been lost and gained many times. And so this group up here looks like it acquired this island, but then there's no simple explanation. There can't be a single gain or loss event that explains this pattern here. So, if there was an ancestor here that acquired the island, then it must have been lost multiple times. If there was an ancestor, here, then it still has to be lost multiple times, or you have independent gains. And so the evolutionary history of this is quite complicated, and it doesn't look like a single loss or gain event. However, the presence of it strongly predicts pathogenesis. And so we screened through as many isolates as we could get our hands on within this clade. And the island nearly perfectly predicts pathogenesis. There's one exception here, DF41, that has the island, but it looks like it has a mutation in syringopeptin. And that so probably explains why it's not a pathogen on its own. So we wondered if this is sort of a case where commensals are obtaining a pathogenicity island, and that is driving this lifestyle transition from commensal to pathogen. So while Ryan was working on this project, a really beautiful study by Jeff Chang's group at Oregon State came out showing that Rhodococcus pathogens, through gain of a single virulence plasmid, so Rhodococcus has linear plasmids, by obtaining a single virulence plasmid, commensals on Rhodococcus could rapidly transition to pathogen. And then they could actually lose this plasmid and be commensals. So these are plant-associated bacteria that through gain and loss of a single plasmid can transition from pathogen to commensal. And so we wondered if the same thing was happening in Pseudomonas, but through gain and loss of an island rather than a plasmid. And so Ryan asked if there were any other genes that correlated with lifestyle, or whether these pathogenicity islands were the strongest predictors. So when he did this, he found something really surprising, which was that, so this is our pathogenicity island here shown in red. He found two other islands that very strongly correlated with pathogenicity. So we've annotated these as putative pathogenicity islands 1 and 2. We don't know the function of these yet, but they are nearly perfectly correlated with presence of syringomycin and pathogenesis in these isolates. And then even more tantalizing, we found that there was a type 3 secretion island that's nearly perfectly anti-correlated with the lipopeptide island. And we also found DAPG, which is a known antifungal compound. So this is antifungal molecule that's associated with suppressive soils and ability to control diverse fungi in Pseudomonas. And so this is also anti-correlated with sphingomycin, sphingopeptin island. And so not only are we seeing gain or loss of a single island associated with commensalism or pathogenesis, this is associated with gain of multiple other islands and convergent loss of several islands. And so this suggests that over a relatively short evolutionary time with these bacteria, there's multiple gains and losses. So there's genome-wide gains and losses that are associated with lifestyle among very closely related bacteria. And so we think that Pseudomonas is sort of converging on two solutions for plant and fungal association. These islands are always in the same location, and we have evidence that they're being gained and lost through homologous recombination. So we have this lipopeptide island and these two putative pathogenicity islands. And this confers opportunistic pathogens on plants. And I didn't talk about this, but the sphingomycin, sphingopeptin also has antifungal activity. And then there's a second solution where we have type 3 secretion, which is associated with commensalism on plants. So this is a little unusual, but there are some cases with rhizobia that type 3 secretion is essential for mutualism. And then we have DAPG, that's antifungal. And so we think Pseudomonas, again, is converging on two genomic solutions and that they potentially can be cycling through these very rapidly over very short evolutionary distances. And so using comparative genomics, we were able to find sort of the genetic basis or genomic basis of these transitions from pathogenesis to growth promoting strains. And we've used this approach for a number of other questions in the lab that I won't have time to talk to you about today. And these are things like induced systemic resistance and susceptibility and biocontrol of fungi. And so we have a few recent papers using comparative genomics to find the genetic basis of induced susceptibility locus, and then actually sphingomycin and sphingopeptin, again, controlling antifungal activity. So for the last few minutes, hopefully I won't go too fast. I'm going to tell you a recent story that was just published a few weeks ago about how host genetics shape the microbiome. And so this appears to be a bit of a switch in gear, but hopefully if I have time at the end, I can link it back to this diversity in Pseudomonas story. So in plants and animals, dysbiotic microbiomes are associated with disease. So we need a normal microbiome structure to promote health, both of us and of our plant crops. And so it's known that in animals, if you take antibiotics and wipe out your microbiome, it can leave you vulnerable to opportunistic pathogen infections, like Clostridium difficile is a classic opportunistic infection that occurs after antibiotic treatment. And dysbiosis is also associated with autoimmune and other diseases in animals. It turns out that dysbiosis is also a problem for plants. This is a really nice paper from Shengyang He's lab showing that if you have immunocompromised plants, and so this is 3 hormone receptors, so impaired in receptor kinase mediated immunity in leaves, you get a shift in the microbial community that results in overgrowth of proteobacteria. And then these otherwise commensal microbes become pathogenic. So you can see disease symptoms developing from the microbiome. This is also associated with reduced microbial diversity. So you have way less microbes or diversity of microbes in this immunocompromised state. Plants also do something really cool that I think has not really been described in animal literature. And that's that they can seem to enrich for specific groups of beneficial microbes that are associated with health. And so a classic example of this is suppressive soils, where plants sense a fungal or insect pathogen and then can reshape the soil microbiome into one that's protective against future attacks. And so this has been observed probably most commonly in wheat, where wheat can sense a fungal pathogen and then reshape the microbiome into one that is enriched in microbes such as Pseudomonas that can protect against fungal pathogens. And then with microbiome sequencing, we're seeing more and more cases where Pseudomonas actually can form these blooms. So this is a publication on corn from Ruth Lay's lab, showing that they spontaneously had this bloom of Pseudomonas that occurred late in the season, and they don't really understand why. But both of these together suggest that plants may have a genetic mechanism to enrich for specific beneficial microbes. And so we wondered if we could find plants that could either constitutively recruit or impaired in recruitment of Pseudomonas to find the basis of things like suppressive soils and the Pseudomonas bloom. And so this is the work of Yi Song, another postdoc in the lab. And so Yi made use of a previous screen that was done for hormone immunity crosstalk. And this was from my postdoc lab, done by a postdoc, Zucheng Zhang. And the first of these papers was published a few years ago, describing the screen and one gene that came out of the screen. These are specifically a screen for immunity in roots. And so we thought that this would be a collection of mutants that would give us the potential to find new regulators of the microbiome without doing a whole, for a genetic screen of 10,000 mutants, because that's difficult for microbiome phenotypes. So Yi rescreened this collection of HSM mutants, and he did it using a 48-well plate where we have plants that we grow hydroponically. So they have this little mesh disc on the surface, and then the roots grow down into hydroponic media. And we can add fluorescently labeled bacteria and then measure fluorescence as a proxy for bacterial abundance. And so this lets us rapidly assess Pseudomonas level in the rhizosphere. And so using this, Yi found a few plant mutants that either have decreased or increased levels of Pseudomonas. And he chose to focus on HSM13, which has massive overgrowth of Pseudomonas in this reductionist system. And so one of the first things that Yi did was ask whether this HSM13 mutant also has increased Pseudomonas in natural soil. And so he planted HSM in natural soil and asked whether it has increased Pseudomonas. Pseudomonas fluoresces on iron-limiting media. And so it's easy using a culture-based approach to see how much Pseudomonas is there. And he found that HSM13 has about 10 times more Pseudomonas in the rhizosphere than our wild-type genotype Columbia, and much more than bulk soil. So it seems that HSM13 enriches for Pseudomonas in the presence of a natural community as well. And so we sequenced the microbiome of HSM13, and we found actually that it doesn't look dysbiotic, like the example I showed you from Shengyang He. We don't see this massive overgrowth of proteobacteria, although there is definitely, by principal component analysis, there is a shift between HSM13 and wild-type plants. And it turns out that most of that shift is because of increased Pseudomonas. So looking at diversity or taxa that are significantly different in HSM13, it's largely limited to the family Pseudomonidaceae. Although we do see reduced diversity. So the HSM13 mutant has reduced diversity compared to wild-type plants, which is similar to that dysbiotic mutant and is often associated with dysbiosis. So to determine if HSM13 is dysbiosis, we did microbiome transplants. And these work by growing wild-type plants or HSM13 in natural soil, removing the plants, and so then leaving behind the microbiome that's shaped by these communities or by these plants, and then replanting wild-type plants in this soil. And so then we can ask if the HSM microbiome confers benefits to wild-type plants differently than wild-type. And so when we did this, we found that indeed wild-type plants planted in HSM13 soil had a significant growth promotion compared to wild-type plants planted in wild-type soil. And this correlates quite well with an increase in Pseudomonas fluorescens. And so it seems that the HSM13 microbiome is not dysbiotic, but provides benefits to plants. So Yi mapped HSM13 and found that it encodes Feronia kinase, which is a master regulator of plant immunity and development. And so I'm sure many of you have come across Feronia and other areas of plant biology research. Our allele has a point mutation in the kinase domain of Feronia. And then this is just genetics showing that if you cross HSM13 to FUR-4, which has a transposon insertion, here it doesn't complement, but if you cross it to wild type, it does restore Pseudomonas growth, indicating that it's recessive. And then the other Feronia alleles behave very similarly to HSM13. And so Feronia does a lot of things. And so there's a few ways that it is involved in immunity that we thought could affect Pseudomonas growth. So it can interact with ROP-GEF, ROP2, which activates NADPH oxidase to make reactive oxygen. And so we thought maybe this could inhibit Pseudomonas growth. It also inhibits MYC2, which regulates jasmonic acid signaling. And so we thought maybe through JASA antagonism, increased JA could result in decreased salicylic acid, which could affect Pseudomonas growth. And Feronia also interacts with some of the receptor-like kinases like BAC1 and FLS2 to control pattern-triggered immunity. And so because we have mutants in all of these pathways, we were rapidly able to test whether any of them are important. And so it turns out if you lose jasmonic acid signaling or pattern-triggered immunity, it doesn't affect rhizosphere growth of Pseudomonas. So this is different from leaves. However, if you lose reactive oxygen production through NADPH oxidase, we saw an increase in Pseudomonas fluorescens, partially phenocopying this is FUR-8 because there's so many Feronia alleles. HSM13 is FUR-8. And so if you image reactive oxygen in the Feronia roots, you can see that this is a reactive oxygen binding dye. There's decrease in Feronia, and then Pseudomonas actually further decreases reactive oxygen levels. And so it seems that Feronia likely is working through this NADPH oxidase pathway. Because I'm getting a little short on time, I'll quickly go through a few of these slides. So we can overexpress NADPH oxidase and reduce level of Pseudomonas in an HSM13 background. So by overexpressing this pathway, we can actually rescue the loss of activation of this pathway in HSM13. And so we think that this is the pathway that Feronia is working through. And so coming back to the question of how Feronia or how plants might sense the environment to enrich beneficial microbes in the presence of a fungal pathogen, it turns out that fungi can make something that looks a lot like the ligand for Feronia. So fungal pathogens and nematodes can make Ralph-like peptides, and Ralph peptides are the ligand for Feronia. And they've been reported to allegedly do this to suppress jasmonic acid signaling in order to repress resistance to fungi. And so we thought maybe plants were guarding this pathway. So by sensing Ralph peptides could also recruit Pseudomonas as a second line of defense against fungal pathogens and nematodes. And so Yi added purified Ralph peptides or overexpressed a Ralph peptide in plants. And we found that indeed addition of Ralph peptides is sufficient to recruit Pseudomonas in a Feronia dependent manner. And so we think that maybe when plants sense fungal pathogens, they could increase Pseudomonas as a way to protect against future attack. And so that's described here, where sensing a Ralph peptide results in suppression of JA signaling, resulting in plants being more vulnerable to that fungal pathogen. But at the same time, plants could also repress root basal levels of reactive oxygen to allow proliferation of Pseudomonas. And so I think I'll stop there. I had a couple more slides, but I think I'm low on time. Collectively, we think that by understanding how plants can recruit beneficial Pseudomonas and then how Pseudomonas provides these benefits, we can facilitate things like microbiome engineering. And so hopefully we can join plants with other microbes that can provide benefits other than nitrogen fixation to help protect plants in a changing climate. And so with that, I'd like to thank the people in the lab. So I tried to acknowledge people as I went, and here are our funders, and Shengyang He was a collaborator on the Feronia story, and I'd be happy to take any questions.